bioeconomy and sustainable food chain, which is operating under the umbrella of European Circular Economy Stakeholder Platform. And usually we are meeting and discussing different topics regarding food waste, regarding biomass effective use or converting into different product. But today we are meeting to discuss completely different topic, which is called bioremediation. As much important as another one. We are also a member of the Horizon Europe project, which called NIMFE, and the main goal is to improve the bioremediation technique solution and implement it into environment. So before we start, I would like to ask you, the audience, how familiar you are with the bioremediation topics, bioremediation solution. So Lucas, could you display the first questions and please answer if you know the topic which we're discussing today. So I don't know, can you hear me? By the way, yes?
Yes, we can hear you.
Okay, because like So Lucas, could you display the question?
I see that the question has been displayed and answers are arriving.
But I think we can't see it. I don't know. Have you seen it? I don't think. At least I'm not seeing.
Yes, I saw it and I could vote.
Okay, so I don't know if we can see the answer now.
74% said yes, I'm familiar with this application.
So most of you like heard about the topic. So today with us we have like multiple experts which like dealing on everyday basis with the bioremediation research. So we will discuss during our talk about the different techniques, different solution, also how we can really scale up the bioremediation in Europe. Firstly, maybe I ask Giulio Zanaroli, Associate Professor from University of Bologna. He's also a coordinator of the NIMFE project, to tell us a little bit more why we need bioremediation and what really bioremediation means. So Giulio, please, if you can start your presentation.
Yes, thank you, Agnieszka. I'm sharing my screen. Hopefully you see my presentation.
Yes.
Okay, yes, I will give a very short, let's say, introduction on bioremediation, and I will take the opportunity also to illustrate shortly how the NIMFE project is trying to address some challenges related to this topic. Some of this information may be not new for the 73% that has already answered that they know about bioremediation. Anyway, as you all know, chemical pollution is a major ecological problem that is causing serious harm to human health and the environment, not only because of toxicity problems, but also because of reduction in biodiversity and ecosystem functioning services. And it is also a quite complex problem because actually we are experiencing a large variety of chemicals that are released into the environment from even larger number of contamination sources and that are affecting almost all environments, all environmental matrices from soil, groundwater, but also surface and sediments both in terrestrial and marine environments. And actually, bioremediation is, let's say, an approach or a technology that is believed to be promising and sustainable in tackling such an environmental contamination. In particular, bioremediation consists in processes that use living organisms, mainly microorganisms such as bacteria and fungi, but also plants and other organisms, to degrade or detoxify contaminants. And this can be not only applied to degrade or remove the pollutants that have been released into the environment and that have accumulated and are persisting in contaminated environmental matrices, but can also contribute in reducing the amounts of pollutants that are released to nature, for example, by improving wastewater treatment in order to, let's say, avoid some, for example, micropollutants are released into the environment. One of the main aspects of bioremediation is that, of course, it promises some benefits compared to the more conventional physicochemical approaches, since actually it is a bioprocess that is taking place under mild and life-compatible conditions, for example, ambient temperature, without the use of highly reactive chemicals or solvents. And this turns into the absence or, let's say, the production of minimal alterations in the physical, chemical, and biological features of the treated matrix. For these reasons, bioremediation is considered a nature-based approach that not only can remove, degrade pollutants in environmental matrices, but that can also promote the recovery of the biodiversity and the revitalization of the sites after remediation. The main actors playing in bioremediations are microorganisms, which are very small living organisms that have some peculiar features that makes them particularly interesting, since they have very often a high metabolic versatility, so they can use and degrade a large variety of different chemicals. They are fast growing, and they typically are characterized by a fast natural evolution or adaptation, which is making these microbes evolving through and acquire degradation capabilities. Another important aspect is that they are ubiquitous, so they are inhabiting all environmental niches and also higher organisms, typically in the form of complex communities. Of course, their degradation abilities depend on enzymes they are producing, which are the catalysts that make possible, that make reactions happen rapidly in these biological systems. Other important actors in bioremediation can be also higher organisms, in particular plants, that, for example, have the natural ability to extract and to process chemicals that they are using as nutrients from the soils and water in which they are living. This is typically done also in close cooperation with bacteria, but also some animals can contribute to bioremediation processes. For example, plants can act as biofilters and or absorb pollutants in water, whereas earthworm can promote soil aeration and therefore microbial activities. In general, the main strategies used in bioremediation are consisting in the control on the environmental conditions in the contaminated matrix and in the supplementation of the suitable nutrients to promote the activity of the degrading microorganisms, the approach known as biostimulation, or also in the introduction of specialized degrading microbes, which is referred to as bagmentation. So, of course, the situation is very complex. Real polluted sites are presenting a high degree of complexity, which makes sometimes bioremediation not fully effective. And these bottlenecks are related, for example, to the characteristics of pollution. We might have multiple contaminants, so we might need different micro-organisms in order to degrade these high complex mixtures. Pollutants may be present at very low concentration, so very hard to be degraded, or at low bioavailability. Other important factors are related to the environment or the matrix that is contaminated, for example, because of the lack of specific nutrients such as electron donors or receptors, or non-optimal physical-chemical parameters. Another important aspect is the fact that these degrading organisms sometimes lack of competitivity. So the degrading microbes are not able to persist and grow in the contaminated matrix, or, for example, they need a long acclimation period once they are introduced into the contaminated matrix. There is, of course, the possibility to improve all these aspects, and here I'm presenting very briefly how these challenges are proposed to be addressed in the European project NYMPH that has recently started, which actually is proposing the use of association of multiple biologics that cooperate in the degradation of single pollutants or complex mixture of pollutants, but also the development of tools to improve bioremediation efficiency of these organisms, both at the system level or at single biologics level. And an important other aspect is the use or the development of model-based microbiome management strategies that will be addressed in a talk later, in order to make these complex communities of microorganisms work in a more efficient way for the degradation of pollutants. So very briefly, the NYMPH objective is to develop not only bioremediation, but revitalization strategies for contaminated matrices that are based on systems consisting of assemblies of multiple biologics, and of course to assess them from a number of points of view, both, let's say, not only in at laboratory scale, but at field or pilot scale for real contaminated sites. In particular, we are addressing 4 sites that are representative of very common polluted situations all around Europe and the world. An industrial site where the groundwater, the aquifer sediments, and the soil matrices are contaminated with petroleum hydrocarbons and chlorinated aliphatic hydrocarbons, but also by some heavy metals. An agricultural field that is contaminated with microplastic and pesticides. The hyporheic zone, so in particular at River Erpe, the sediment and the surface water that are contaminated with pharmaceuticals released by a wastewater treatment plant, and municipal wastewater, in particular in Spain, a municipal wastewater in Spain, which is contaminated with pharmaceuticals and microplastics. In the project, we are also proposing to develop some genomically edited biologics to be tested, and this, of course, needs the use of facilities where these biologics can be tested in confined environment, which is a 5th project site. So in general, the approach is a tiered approach. In a first tier, of course, we need to select, isolate, and characterize a high number of biologics that are suited for high performance degradation of the pollutants that are characteristically occurring at the 4 contaminated site. Then the approach consists in assembling multi-component systems based on different biologics that are introduced in the real contaminated matrix from the project sites and tested for their ability to degrade the target pollutant mixtures. In this step, a crucial aspect is also the modeling of the microbial community, of the microbiome of the system, in order to identify which are the keystone species and which are the metabolic activities that need either to be enhanced, complemented, or suppressed in order to boost the bioremediation activity of these organisms. In a second tier,
Giulio, I think slowly we have to kind of come to the end. Yeah.
We will also develop or improve some biologics, and then the most important thing is that all these systems needs to be tested at pilot or field scale and to be assessed in terms of technical, environmental, economical, and societal points of view, which is, of course, the most relevant part also to prove the scalability of the bioremediation approaches. So I conclude here, and apologies for taking a bit longer, and thank you for your attention.
No, it's okay. I understand that it's your passion, but I will be a bad policeman here because we have like a lot of great experts, you know, who can probably take for ages. So I have to have a time limit. So as you see, bioremediation could be effective tool for cleaning environment. So let's ask you the next question. How concerned are you about pollutants and the impact on the environment, especially soil and water? So Lucas, could you display the second question? And then we will slowly move to the policy aspects, because we have representative of the European Commission and European Economic and Social Committee. So I warmly welcome Dr. Silvia Maltagiatti, Policy Officer, Circular Economy and Bio-Based System from the Directorate Research and Innovation. And we also have with us Kilian Lohan, member of European Economic and Social Committee. So you are representative of the policy side, and as I know, there is already a few projects regarding bioremediation going on in Europe. So I see that European Commission are concerned and see the future in this solution. So as I see the answer, we are mainly very concerned. I think no one answered they are not concerned, which is good, and maybe more or less happy about it that we are concerned. So please, Silvia, could you now tell us a little bit about the policy and policy angle towards bioremediation topics?
Yes, thank you very much. I hope that you can hear me correctly.
Yes, we can.
So first, good morning, and thank you very much for the invitation and for the opportunity to meet you today. Actually, more than policy, I would say that my talk would recap some of the different areas in the programs supporting R&I with European funds where remediation and bioremediation are covered. So I would like to ask Lucas if he can please share the slides that I prepared, and there are many, but we will go through them very quickly, and because many of the things that are included are well known, but I would like you, in fact, basically, as said, to give you a picture of the EU funding. So you see that I have included in the first slide also the name of my colleague Thomas Kalikowski from the same unit as mine, so Circular Economy and Bio-Based Systems in DG Research and Innovation, as he's actually the policy officer for the projects Biosismo, Symbiorem, Nymphe, and Mibirem. So you may know him as well. So next slide, please. The slide visualizes, as said, the different areas in the EU R&I programs that can contribute to supporting the R&I and bioremediation solutions. Basically, the framework program Horizon Europe with its cluster 6 on food, bioeconomy, natural resources, agriculture and environment. Then the missions under the same Horizon Europe, such as a soil deal for Europe and restore our oceans and waters. And finally, the Circular Bio-Based Europe Joint Undertaking, which is a partnership between the EU and the Bio-Based Industry Consortium, worth 2 billion euros, running from 2022 to 2031, and aiming at supporting innovative bio-based solutions. Next slide, please. About Horizon Europe Cluster 6, the Strategic Plan 21-24 envisages as expected outcome the prevention and removal of pollution, with in the table below with impact areas on enhancing ecosystems and biodiversity while turning air, water and soil into clean and healthy status. Next slide. So as Cluster 6 as 7 intervention areas, we will see that many of them, in fact, deal with basically pollution and remediation. Number 2, intervention area number 2 is biodiversity and natural capital, where the overall goal and R&I broad lines are explained in the Horizon Europe specific program 21-27. In particular, this area is about understanding biodiversity and ecosystems, the multiple services they provide, and the concept of planetary boundaries, as well as developing solutions harnessing nature's power and complexity. And one R&I broad line is especially addressing adaptable and multifunctional nature-based solutions, addressing challenges in cities, rural and coastal areas related to climate change, natural disaster, biodiversity loss, ecosystem degradation, pollution, and citizens' health and well-being. In fact, under this intervention area, we find calls in the annual work programs, next slide, please, such as the call for a research innovation action on biodiversity economics and finance, unlocking financial flows towards reversing of biodiversity loss, aiming at, among other outcomes, to reorienting financial flows towards activities that benefit protection, restoration, and sustainable management and use of biodiversity and ecosystems. The call closed last 28th of March. Next slide. We have a second call under intervention area 2 again. It's another research innovation action in 2024 on reintroduction of landscape features in intensive agriculture areas, which will be presented sometime during the Info Days next 28-29 September. Next slide, please. The second intervention area is number 6 on bio-based systems, for which the overall goal is to enable the transition away from a fossil-based economy by encompassing the sustainable sourcing, industrial processing, and conversion of biomass from land and sea into bio-based materials, products, and processes, including from industrial biotech. And a specific R&I broad line, again in the Horizon Europe specific program, addresses the use of biotechnology for application in competitive, sustainable, and novel industrial processes, environmental services, and consumer products. In fact, under this intervention area, we find calls in the annual work programs, next slide, please, such as the call that you know very well for this research and innovation action on environmental services, improved bioremediation and revitalization strategies for soil, sediments, and water, that you know very well because it gave birth to the projects Nymphe, Symbiorem, and Biosismo. And then the call, next slide, again, that you know very well, for an innovation action on microbiomes for bio-based innovation and environmental applications that funded the project Symbiorem. So changing scenario, we find that emissions, the first emission, restore our ocean and waters by 2030 with 3 objectives and corresponding 9 targets. Next slide, please. Among which there are specific marine nature restoration targets, including degraded seabeds and coastal ecosystems, which has led to the selection of world programs. Next slide, please. Including innovation action to prevent, minimize, and remediate chemical pollutions. We find 2 projects, the Remediation Responsible Hub for long-term governance to distress the Mediterranean Sea from chemical pollution, and eMermaid, innovative solutions for Mediterranean ecosystem remediation via monitoring and decontamination from chemical pollution. That, in fact, could be also part of the discussion today. The second mission, next slide, a soil deal for Europe, has 8 specific objectives. next slide, including to reduce soil pollution and enhance restoration, which has led to include in the work program 2022 a specific call on that, next slide, for a research and innovation action on remediation strategy, methods and financial models for the contamination and reuse of land in urban and rural areas, funding 3 projects, Aragorn, Edafos, and Islander, that again could be also part of the discussion today or of the next, say, meeting. Next slide. To let you know that the last part of Horizon Europe covering 25-27 will be based on the new strategic plan already published in its first draft. And it has identified some gaps to be covered in the next work program in many sectors under agriculture, forestry, and rural areas. For example, there is a need for soil decontamination. Under seas, oceans, and inland waters, we should focus on restoration methods for European lakes and freshwater ecosystems. Under the bio-based innovation systems, again, more will be done for restoration, for pollution detection and remediation. And last, the circular system will include actions addressing pollution challenges, especially in water bodies. Next slide, please. Finally, to come to the last EU mechanisms of today, delivering funds to support R&I for bioremediation is the Circular Bio-based Joint Undertaking. As said, this is a partnership, next slide as well, a partnership between the European Union, represented by the European Commission, and the Bio-based Industry Consortium, BIC, providing 2 billion grants to projects advancing competitive circular bio-based industries in Europe. And the CBJU operates under the rules of Horizon Europe and for a decade. And there is a document called the Strategic Research Innovation Agenda, setting the scope, the governing boards and the advisory bodies, the financial aspect and the delivery mechanisms. And in fact, we already delivered work programs with calls for grants in 2022. The work program was published, granting 120 million euros for projects. The 2023 work program already published, granting 215 million euros, and 2024 indeed is under preparation. Next slide. And within the Strategic Research and Innovation Agenda, there are specific priorities are set, among which one to protect and enhance biodiversity and ecosystem services, and which has led the EU Mission on Soil to publish a kind of paper where the synergies between the Mission on Soil and the joint undertaking are cleared. With common goals such as soil pollution restoration and others. And next slide. The joint undertaking has already confirmed this focus, publishing in the Work Programme 2023 a call for a research and innovation action on phytomanagement, curing soil with industrial crops, utilizing contaminated and saline land for industrial crop production, that you may have seen, of course, the call deadline is next week. So I hope that you have seen it before. More calls on remediation and indicators and bioremediation are planned in the future. Maybe not in the next work program, but for sure it will come more and more. To conclude, I also want to present in the next slide that the annual work programs of the JU always include, apart from the specific requirements that proposal must comply with, including on demonstrating that the provision of biomass for the bio-based industries contribute to all the environmental, say, objectives. So, for example, any biomass crops that can demonstrate the contribution to remediation and restoration of soil is in the scope of the JU. And with that, I have concluded. Thank you for the attention, and I hope that at least this can provide an overview of the current and future trends in R&I funding programs. Thank you.
Thank you very much, Sylvia. As we see, we have like a lot of opportunities to find the funding for the future project. So that's great. So now I give the floor to Kilian Lohan from European Economic and Social Committee. Good morning, Kilian.
Good morning, and thank you for having me here, and thank you, Sylvia, for all that information. Wow, there's so much happening in this space, and such an important topic for us to have an EU circular talk on. So my name is Kilian Lohan, and I'm a member of the European Economic and Social Committee. And as a member of that, I've worked on circular economy topics for a number of years now, including writing an opinion that led to the establishment of the European Circular Economy Stakeholder Platform. And many of you will already know in this audience that this platform is a one-stop shop for circular economy in the EU, and indeed with some neighbouring countries also. We publish regularly through social media, sharing information and knowledge through Twitter and LinkedIn, but also by producing a regular newsletter and posting information and knowledge sharing on the ESESP website, the European Circular Economy Stakeholder Platform. It's about a platform that is about exchanging ideas and best practice, but also importantly, it is led by stakeholders like you, and it is an opportunity to feed into policymakers where barriers are identified to transitioning to a circular economy. So it's become a very valuable space for talking, and indeed these EU circular talks, as organised by the leadership groups, are a very important element of having these conversations. And I think importantly for these types of conversations, which are so familiar to us now, particularly online discussions, I think the real added value with these EU circular talks and today is that there's a tangible output at the end of the process, and we have something concrete to move forward with after the event. So I want to thank you all for your participation, and the program for this morning looks really interesting, with really expert speakers here at the EESC. Our contribution is as a consultative body to the legislative institutions within the EU, and in that regard we make major contributions in terms of climate and sustainability and circular economy through writing our reports, which are called opinions, but also through sending delegations to the different international conferences. And in that sense, we have been pushing the idea of nature-based solutions for a number of years now in the space of circular economy, but also in biodiversity and in climate conversations. And I see many very strong practical examples of that being shared with us today, and I know more to come in the later panels. One of the propositions that we regularly put forward in our opinions is the idea that there is more synergy needed between different policy areas such as bioeconomy, climate, biodiversity, and circular economy. And I would like to welcome and recognize the fact that the ESESP leadership group on bioeconomy really leads by example in showing where these synergies can occur. And indeed, I think in what Sylvia just presented in the research area from the Commission, we see regular attempts to try and amplify those synergies and support research in those particular areas. We would love to see that applied across all the different DGs in the Commission and all the different spaces that this works on, but certainly research is a leading light in taking that particular approach. And that idea of policy coherence, I think, is particularly relevant to bioremediation, because we see how many different areas it cuts across, from environmental policies to agricultural policies, circular economy, indeed even in water and in research. And we see that in some of the opinions that we've produced here at the EESC. In our soil strategy opinion, for example, we specify the role of bioremediation, and also in biomass production of algae in the context of the blue economy. The ESC for the first time produced an opinion this year on the algae initiative. So you will see strong examples of practices, best practices some that are shared with you today, but if you want to go to the ASSESP website, you will see there that we share best practices, some very interesting ones. There's one, for example, on ashtrays that are being produced called Purei fungi, using fungi and mycelium in order to create ashtrays that actually perform a bioremediation function. So I know there's much more of that coming in the next panel, and a very fruitful discussion coming this morning. I want to thank you for your participation and your time and the work that everybody's doing in this space. And thank you, Agnes, for inviting me today and giving me the space to say a few words.
Thank you very much. It's a pleasure to host you here, absolutely. So before we go to the deep dive into bioremediation technique, I would like to ask you another question. Do you believe that the bioremediation of used living organisms, bacteria, plants can be effective solution? So do you think that it's really could be effective and efficient? So please, Lucas, if you can display a question. And I maybe add that, of course, you also can ask us a question. So there is a chat. So please feel free any moments if you feel concerned, if you have a question mark in your head, just write a question, because in the second part of our circular talk, there will be panel discussion with our great experts. So they will try answer to all your concern. Okay, so I see that mostly you believe in bioremediation. There is 70%, and there is no answer. Oh, we are very kind of full of hope. No one thinks that there is no effective. So yes, maybe and yes. I think all of you rather supporting the bioremediation method. So now we go to the session where we would like to show you the different techniques which are used in bioremediation, because it's not only just implement the bioorganism and they make a job. There is like a special different techniques which make it happen. So first, I would like to introduce Professor Marco Candela from Department of Pharmacy and Biotechnology, University of Bologna from Italy. And Professor Candela will show you and tell you a little bit about biomodeling microbiome solution. So please, Marco, thank you for joining. I know that they are busy today, so that's why. Please, okay. The floor is yours.
Yeah, thanks so much. Thanks everybody, and good morning everybody. So I'm going to share the presentation. Just a second. More. So it's here. It's coming.
Yes.
So do you see the presentation?
We see all. Yes, we see it now. Yeah.
Okay. So thanks again. Just what we are talking about. We will talk about, of course, microorganisms, and in particular about metagenomes and microbiomes. So the point is that in nature, the microorganism doesn't live as a single species. Indeed, they live in a complex community where single species work together for the provision of a given set of ecological services. And this complex community are the microbiomes. And so we have microbiomes in the air, we have microbiomes in the water, we have microbiomes, of course, in the soil, and microbiomes lives in association with a complex organism, are part of complex organisms. So there are microbiomes that lives in association with plant and with all the animals that are populating the planet. So this microbial ecosystem are considered nowadays a kind of life support system for the total biosphere. So how we can study this microbial ecosystem? so it's not good. Okay. The only possibility to study the microbial ecosystem is by metagenomics. So it's by molecular approaches for sequencing their DNA. So the DNA that is extracted from this complex community of different microorganisms. So essentially, for a given ecosystem, we simply sample the ecosystem, such as the soil. We extract the total DNA from this complex matrix, and then we sequence all the DNA which is present there. And this way, without culturing the microorganisms, we can understand which type of microorganisms are present, which species are present, at which relative abundance the different species are in the ecosystem, if they have an even or uneven distribution, and also we can know the functional potential of all these microorganisms, what they are able to do, and how they interact for the provision of the selected ecological services. So how they interact for providing benefit to the ecosystem, such as the soil or the host where they are associated to the host. So the interaction between different microorganisms that populate a microbial community is the focus of this talk. Since microbes from different species can interact within a community of microorganisms in different ways. There is, of course, the competition where different microorganisms that populate in a community, different species, can interact, can compete for the same resources, but there is also the syntrophy, when there is a kind of connection in which what is produced by the metabolism of a first microorganism became a resource for the second one. And otherwise, there is the cross-feeding that is highly important, is when a given species feed a second species. Okay, so these are three types of interaction that can happen in a complex community within all the different species that are present in community, and which is so important for the provision of the ecological service. So the ability that the community at all is able to provide competition and to cooperative behavior, which is the syntrophy, when a metabolic endpoint from a species is the substrate from the second species, and the cross-feeding, when one microorganism feeds the second microorganism. So there is another way of competition that is called the public goods that happen when a microorganism, a given species in the community, is able to digest a complex polysaccharides, for instance, in the extracellular environment, in the monomer or dimer. This monomer or dimer are the substrate of the first microorganism that was involved in a direct digestion or hydrolysis of the complex polysaccharide, which is outside the cells, but can also feed a second microorganism, which is not capable by its own to digest the complex polysaccharides. And so the enzyme, the external enzyme, is a glycosidase, which is produced by the first microorganism to create the monomer that feed both the producer of the enzyme, but also the second microorganism that is not able to produce the enzyme by its own, is called public good. And this is another important point about the ecology of interaction of different microorganisms within a community. So we can see a complex microbial community as a complex market in which different species interact by the production of substrate, the sharing of substrate. So it's kind of trading, of molecular trading, that put connection between different microbial species in a complex community. So the point is that a microbiome is made of hundreds of different species that interact in this complex way, that establish species-to-species interaction of different kind and different intensity. So the only chance to figure out the interaction process between the different species that populate a complex community is to apply the network theory. The network theory is a topological representation, a mathematical model that allows to figure out the different interaction between different microorganisms within a species complex. And here you see a very simplified version of the network in which the dots are the different species and the connection are to visualize the type of interaction between the different species in the community. And so you will see here that are some species that are marginal in the community and some other species that are more central in the community, so are more connected with the other species. The more connected species are called keystone species, so are the microorganisms which are important for the creation of the entire nectar structure. Without that microorganism, we won't have the network, and without the nectar, we won't have the community of interactive species. So here you have some type of network that can be obtained when you study a microbial community that has a different topology in terms of modularity, number of different modules, in terms of sparsity, in terms of number of degree of a positive and negative association. Positive association are a cooperation between two microorganisms. A negative association is an interaction, but is of competitive nature. So you have a different layout, different topology from the different network in terms of type of interaction between the different species, and so the network can acquire different structures in terms of modularity, degree of connectivity, and degree of positive and negative association and sparsity. So there is a topology of the nectar at all, and for a given species, you know how this species interact with the other species, and you also know which are the most important species supporting the nectar structure that are the ones that are much more connected respect to the other and are called keystone species. Another important characteristic of the nectar are the hubs. And you see the hubs are this type of nectar modules are highly visible here in this representation that are a group of highly interactive species that are grouped around a specific keystone species that support all the partner species in the hub, and sometimes a given hub in the communities is connected with the given functionality. And this is a very important point. So why is it important to know the network structure where you study a microbial ecosystem, and the microbiome ecosystem in particular? So when you study different species that are interacting for doing something for the provision of a functionality. And here you have a simple cartoon. Here you have the different microbes, and then another level of information can be to know the name and the functional potential of each of the microorganisms that populate the ecosystem. But a deeper level of information is to know how they interact. So how these different species interact each other. And this is the ecological modeling, and this is allowed thanks to the network theory. And this is very important, this type of information. So how the different species interact in the context of the network is highly important for the modulation, for a knowledge-based modulation of the ecosystem function. Indeed, if I know how the different microorganisms interact each other, I can select some I can select some target for a modulation of the ecosystem and propagate the modulation effect in all the network microbiome network. You see here, I can modulating the entire set of microorganisms that are involved in a given set of functionalities, I can just modulate just 2 microorganisms involved in the network and then propagate the modulation across the entire networks. So with this type of information, I can eat 2 microorganisms to eat a microbial hub and eat a group of microorganisms that together provide a specific functionalities of interest. So here is another visualization of the same story. For instance, if I modulate a microorganism that is peripheral to the network structure, I can only increase the functionalities that are due to this peripheral microorganism. If I modulate a microorganism that is central to the network structure, I can increase the functionalities that are due to this microorganism, but also the functionality of all the other microorganisms in the network that are connected with my target. So I can propagate the modulation activities across the network, selecting the hubs, the group of microorganisms that are responsible of a given functionality of interest for me. So how we can create networks is very easy, because networks can be created just constructing a correlation matrix. And so by raw data of the quantitative distribution of the different microbial species in different samples from a given ecosystem, I can create a correlation matrix and see which type of microorganisms are positively correlated and so interact in a positive way, and which type of microorganisms are negatively correlated and so interact in a competitive way. And then, basing on this correlation matrix, I can create a mathematical model of the network with all the connection between the different microorganisms, as we have seen before. There is another type of network we can create.
Just Marcus, slowly, we have to finish.
I'm closing. There is another type of network that we can create that is not based on the correlation between the different microorganisms, so it's not a mathematical model, but can be created basing on the genome scale metabolic model. So I can create a model of a microorganism with respect to their genomic and the biochemical capacity. And basing on this model, I can create a model of the interaction between 2 and more different microorganisms. So I know the microorganism, I know the genome, I know his biochemical propensity, and basing on this information, I can know how the different microorganisms interact in an ecosystem. And this is very important. It's based on complex matrix of interaction, but is very important because by using this method, I can get a community. I can give a given substrate to the community. I see in the community which type of microorganism interact in the metabolism of this substrate and which are the endpoint of this metabolism. So this is a virtual model that represents the total knowledge of the behavior of a community with respect to the digestion, with respect to of a given metabolism, but with respect also to the biotransformation of a given substrate or a given xenobiotics. Thanks so much.
Thank you very much, Marcus. And if I may make a small change in the program, Marcus, if you allow me, and before we go to the bioremediation, we stay in bacteria world still, and maybe we should talk about the bioremediation. So I would introduce Dr. Narcis Pous, or we have Sebastian, because there is also a change of the speaker. Yes, we have, I think, Narcis, yes, with us.
Yes,
so
I apologize
Dr.
for Sebastian.
Welcome warmly, Dr. Narcis Paulus from Laboratory of Chemical and Environmental Engineering from Universitat de Girona. So please tell us a little bit more about the electro-bioremediation. What does it mean exactly?
Yeah, I'll give you. Okay, I hope everything, all of you can see it. I will talk to you about this concept, electro-bioremediation. Why we need to study new technologies. As you know, in bioremediation, we started first with a dilutionist solution, and then during the last years, we have different technologies that are able to treat some pollutants, but they require a lot of resources in terms of energy and also chemicals. And the thing of a study of electro-bioremediation is because we want to develop more sustainable solutions that can be cheaper in terms of ecosystem usage and also in terms of money. And for electro-bioremediation, the most important thing is the microbial, what we call microbial electrochemistry. So we use a specific kind of microorganisms that are able to use electrodes as an electron donor or acceptor. It means that it's a niche of microorganisms, but it also means that the process that we are studying are very specific and targeted to the pollutant that we want to treat. Just for you to understand a bit more about these bacteria. These bacteria are not a super different bacteria, but they are available almost around in the world. We can find them either in animal gut or also in paddy rice soils and also in groundwater, especially in places where there is iron in their rocks. Because these bacteria in nature, basically, they are working with iron, oxidizing and reducing solid iron minerals. And when we put them in a reactor with an electrode, they use the same mechanism to oxidize and reduce pollutants. Here there is a photo of an image of a microorganisms. In the case, they are performing an oxidation. Basically, the bacteria is releasing electrons to the electrode. In case they are performing a reduction, for example, the case of denitrification, removal of nitrates, they are taking the electrons from the electrode and removing the pollutant. So we don't need to add chemicals or sparge oxygen to remove pollutants, just a solid electrode, and the bacteria will take the pollutant and remove it. There are several pollutants that can be treated with this concept. Here you can see some. In the left side, there is the electrons that the bacteria that will treat pollutants using an anode, basically, so performing oxidation reactions. Basically, these compounds are organic, and we have some metals like arsenic. On the other side, we also have reductions that are performed in the cathode, where we have much more compounds that can be removed with this system, especially metals, and also some nutrients like nitrate. We performed a project that is called Electra that recently finished. This was a European project that was a kind of competition where different research institutions were developing technologies to remove different compounds from nutrients to hydrocarbons, antibiotics, etc. And from them, we selected four champion technologies that went to pilot. Here are some of the technologies that we studied. We studied using, for example, phytobioremediation combined with electrochemistry. We also had more technological systems where we were removing organic compounds. And also, for example, the case of nitrate, which is the case that I will explain you more in detail. For the removal of nitrate, the concept is relatively simple. Basically, we need to take the water from the well and introduce it in a cathode. In a cathode, where we introduce a specific kind of bacteria, we are able to transform the nitrate into the nitrogen gas in the 5-step reactions called by denitrification. And then every time that we use a cathode, we need an anode. And in the anode, we could have several reactions. For example, we could just oxidize water into oxygen, or we could also oxidize chloride into hypochlorite in order to disinfect the water. The process we performed on the development of this technology in the Electra project was starting at laboratory scale where we evaluated the water matrix effect, and we optimized the system in very different aspects, from the microbiome to the reactor configuration, and also the study with other co-contaminants like arsenite, for example. And after this process, we were selected as one of the champions technologies, and we tested the technology on site. Here you can see on the right side our pilot that was located in a municipality of 1,000 inhabitants at the north of Spain. And we basically, the system consisted on a softening unit first and then bioremediation reactors. As you can see, these reactors are quite thin. Basically, one of the important points of electrobioremediation is that the bacteria needs to be very close to the electrode, and the reactor itself has to be very well designed in order to avoid the loss of efficiency. After the pilot testing, we compared the data that we obtained in the pilot with the data we obtained in the lab scale. In the pilot is in the upper side, lab scale in the lower side, and as you can see, the performance were quite similar in both scales. In terms of nitrate reduction rate, for example, we were close to 1 kilo of nitrate per cubic meter per day. We were able to remove the nitrate between 80 and 97% of the influent concentration, which allowed us to bring an effluent that had the quality required by the European directive in terms of nitrate and nitrite. And also something very important is the low cost of the technology. The electricity requirement is very low, between 4 and 6 kilowatts hour per kilogram of nitrate. And something very important is that we don't use any chemical to do this treatment, only electricity supply. Due to the fact that we only need electricity, we think that this technology, electrobioremediation, could be very useful as a decentralized treatment, because we could have the pilot everywhere in the wall. Everywhere that we can add, for example, a solar panel, we can have this kind of pilots, and we don't need to transport chemicals to these places. We only need electricity. Thanks to Electra, we are able to successfully transiting between laboratory scale and the first on-site pilot plants, and we could validate the technology. But now, of course, everything is not ready yet, and we are working in the NIMFE project that Julia was explaining before, and we expect to continue developing this kind of technologies. Thank you very much for your attention.
Thank you very much, Narcis. So as you see, one solution is using bacteria, but there's also other organisms which we can use in bioremediation. So I'm warmly welcome Professor Markus Horn, Professor of Soil Microbiology from Leibniz University of Hanover. Good morning, Markus. Sorry for this small change, but that it seems for me kind of more in line. So please, could you tell us a little bit about vermi-remediation? Yes, this is the topic of your presentation.
Yeah, thanks and a warm welcome to all of you from my side here, and thanks for the kind introduction. So I want to switch gears a little and to introduce a major player in soil that interacts with the soil microbiome. So there's some relation to the first talk that we heard to the interaction of microbes, and I would extend this view by the interaction of the microbiome with higher organisms like earthworms. And the earthworms are the main players in the so-called vermi-remediation. So they are used to remediate organic contaminants in soil mostly, and the only constraints are that the earthworms need to survive in the soils and that the concentrations of the contaminants need to be non-toxic. So recently, in 2020, there was a nice review in Applied Soil Ecology that illustrates the various effects that earthworms might have on pollutants. And if you focus a little on this middle part here of the earthworm picture, you see that earthworms can metabolize contaminants by themselves, either after injection or diffusion of the compounds through the skin into the body. They might bioaccumulate more lipophilic substances. And when you remove the earthworms, you also extract the more lipophilic contaminants. And they might enhance and stimulate the soil microbiome, so to better and more efficiently decontaminate, degrade organic pollutants. This is via the formation of very, very complex structures in soil, so-called drillosphere. You all know earthworms burrow in soil. The burrows represent macropores in soil that really might present aerobic surfaces where oxidation processes go on and are more effective than in the not that well aerated bulk soil surrounding there. And that this holds true, I want to share with you a case study where we researched a little the effect of earthworms on the degradation of methyl chlorophenoxyacetic acid herbicides, short MCPA. And this is a compound degraded under oxide conditions in soil. And there are oxygenases requiring radical oxygen species needed to get the first product, the MCP, which is then easily degraded. So the rate limiting step is really the first step here where the radical oxygen is needed. And to address the earthworm effect, we colonized soil that we brought into the lab with the earthworms. And as you can see on the sketch on the right side, the earthworms form their burrows, and the burrow wall linings are exposed to the oxygen. So is it really that the earthworms stimulate degradation of such compounds? And I can say, yes, that is the case. In this picture, you see the red lines from the earthworm treatments, where we see with the dashed lines the disappearance of the MCPA over time. And in the solid red line, the appearance of carbon dioxide that is derived from this MCPA. So there is mineralization of the MCPA to CO2 going on in soils. And this is much quicker when the earthworms are present than when they are absent, which is depicted with the gray and black lines here. So earthworms indeed speed up the complete degradation of the MCPA to carbon dioxide. And this was related to a huge increase in the internal surface area. On the right-hand side, you see a micro CT, computer tomography picture, with the grayish tubes here reflecting the earthworm channels, the earthworm burrows. And in our experiment, the soil was really reworked by the earthworms, increasing the soil volume by 50% and increasing the aerated surfaces by 300% in the soil. So we really can conclude that earthworms have the potential to increase the degradation of pesticides in soil. And that this was due to a stimulation of the soil microbial community. We had a look at what organisms, what microbes took up and assimilated the C13 labeled carbon of our methyl chlorophenoxyacetic acid herbicide in different depth layers of our soil columns and also in the cast, the droppings of the earthworms that we collected from the surface of the soil. And interestingly, some organisms here depicted in blue, Sphingomonidaceae, and some greenish ones, these Commonmonidaceae, were really prominent in soil and in the burrow walls and the cast to degrade MCPA along with some planktomycetes. So as a microbiologist, we're interested in really getting the key taxa that catalyze a certain process. And in this case, these key taxa were stimulated by the action of earthworms. And then I want to share a second case study with you. This is the degradation of microplastics, in this case made of polylactic acid. PLA is a so-called bio-based, biodegradable plastic, but when you deposit it in soil, it's very, very hard to be degraded by soil microbes. It usually requires a heat treatment before it becomes accessible to the degradation by the soil microbiome. So it is something like pseudo-persistent under environmental conditions. And our question was, could the earthworms eventually help there? And we incubated soil with and without PLA, and with and without earthworms to assess the effect of earthworms on the PLA degradation kinetics in soil. And again, the mineralization of the PLA, the complete degradation to carbon dioxide, was much higher in the presence of earthworms. This is depicted by the dark and light blue line with filled symbols. These two lines differentiate each other because we had different amounts of PLA added to the soil in the presence of earthworms. In the absence of earthworms, only very little CO2 production was observed during the 14 days of our incubation. So the earthworms stimulated the mineralization of the PLA, and we calculated the half-life of the PLA in soil and found that it was less than 10 years in the presence of earthworms, but greater than 50 years in the absence of earthworms. So the earthworms seemed to replace a little the heat treatment which is needed for an efficient PLA degradation and stimulated the PLA degradation, probably via the action of digestive enzymes and likewise feeding and stimulation of their gut microbiomes. So to conclude, I would say bioremediation is a very cost-efficient, eco-friendly, promising bio-based strategy to get rid of organic contaminants in soil, either via direct uptake, via metabolization by the earthworms, or even more important, by activation of the endogenous soil, endogenous microbiota to do the job. So degradation processes are accelerated. The constraints are that the compounds that are in soil are not allowed to exceed concentrations that might become toxic to the earthworms, of course, and that the conditions in the soil that are to be treated are suitable for earthworm survival and colonization. Yeah, with that, I want to thank you for your attention, and I can hand over to Agnieszka again.
Thank you very much, Markus. So now let's go to the water. And I would like to introduce to you Dr. Tiziana Centofanti from Alchemia Nova, Austrian Research Institute. You do a lot of very interesting and exciting project. And Dr. Centofanti will talk about bioremediation in the fresh waters and how we can use mussels, yes, clams, to clean this environment. So please, the floor is yours.
Thank you, Agnieszka. Can you hear me well?
Yes, we can hear you and we can see your presentation.
Okay, first I apologize for not using the other slides, but we also
it's okay.
Have very good slides prepared by Greenovate, which is a partner in this project. I'm presenting Symbiorem, which is a EU-funded project, and I show here all the partners in this project. So all the work I'm presenting today is a cooperation of about a third of the partners you see here. And I'm going to talk about bioremediation of polluted freshwater. In particular, we will look at some initial steps in this project of like how to create a floating wetland in a water system, usually a lake, and creating this type of relationship between mussels, freshwater mussels, and macrophytes. So a very brief overlook at the project itself. So this project is about improving the effectiveness, sustainability, circularity, and also the cost efficiency of bioremediation technologies that target different types of compartments in the ecosystem, like soils, sediments, surface water, and groundwater. So we are developing about 12 new and optimized technologies that utilize also some of the organisms that we just talked about in the previous presentation. So microorganisms, microbiomes, enzyme, fungi, plants, and aquatic animals. We have about 9 different types of contaminated environments. So those are actually our sites where we are operating and installing and developing the technologies that we develop in the lab. And those are scattered across Europe. I'll show you a map in a moment, and also including the United States. We work on soil, surface water, groundwater, and marine sediment. And we're trying to achieve biodegradation rates that range between 60 and 80%, depending on the pollutant. So most of the sites are in Central and Western Europe, also the United Kingdom, and as well as in California. This is a site in Central California in a city called Fresno, which probably most of you don't know, but it's the 5th largest city in California. In Symbiorem, we worked in different parts of the so-called phytomanagement technologies. So we have bioremediation, phytoremediation, and also floating wetland and floating island. So today I will concentrate on these two parts, but mostly on the floating wetland. So I'll talk about these two sites, one in Austria, Lake Neusiedl, and I will just mention Gallions Lake in the United Kingdom. And so these are the study sites that deal with fresh water in Symbiorem. We have the Baltic Sea in Sweden. We have the Lake Neusiedl, and that's what we're focusing on today, in Austria. We also have the Gallions Lake in the United Kingdom and Lake Tolpa in Poland. And at the Gallions Lake, we deal with road runoff and also raw sewage. We have contamination of polycyclic aromatic hydrocarbons, as well as heavy metals and other nasty stuff, and agricultural runoff at the Lake Neusiedl, where we have nutrients, mainly nitrogen, phosphorus, heavy metals, and microplastics. So in this project, we are about to start this part of the project, which is about floating wetland enhanced with freshwater mussels. So what we're planning to do, I don't have data yet because this project started a year ago, but I will show you many of the challenges we had to deal with. But to start with, I'll give you an overview of the plant that we want to install. So this is a system that combines a floating wetland. So we will have a substrate which will be composed of material that is usually upcycled or recycled, as we're so keen in Alchemia Nova to use this type of material. We will work together with University of Wrocław in Poland to develop this type of structure. And on this structure, we will have macrophytes, so plants that live in water. So this is like a floating wetland. And then underneath, we will install a structure where we can hang on to like freshwater mussels. And we have experts at CNR in Verbania in Italy that are currently surveying the sites to see what type of freshwater mussels are already native of the freshwater areas we're working with. And we employ or engage, better said, citizens in this project. So citizen science is a very big component of this project, and also volunteers, not just to conduct sampling and analysis, but also to engage with them, to share knowledge, and to also monitor the sites, as well as decide on this type of structure, for example. So citizens in some of the site will have a say on what type of plants, what type of native plant species we want to install, and how, for example, we want to create this structure for the freshwater mussels. Yeah, so this is a better, let's say, diagram of how this system would actually look like, because you may have asked yourself, how can we really test what mussels are doing here if the system is open? But no, actually, it is not open. We will have enclosured systems where, like, we have mesocosms inside which the mussels are inserted. And so we can have a closed system where we can study how the mussels either absorb the metal or degrade the organic contaminants. So at the moment, we are sampling the sites for the species that we think are going to be there. Definitely, we're expecting the Unio elongatus to be there as a native species, but also we're expecting some other alien species. And so we are also utilizing mussels as a tool to monitor the presence of pollutants at the bioremediation sites, like as a sort of biomarker. So talking about challenges, yes, we had huge challenges, which are mostly related to changes in weather pattern and, for example, due to climate change. So the Lake Neusiedl, and this is now the site that I'm going to focus on in the next few slides, and then I'll be done. In the Lake Neusiedl, we've had historically low and unstable water table. So there is basically no water at the lake, even though this is the sea of the Viennese people, where they used to go for vacation in the summer. They no longer have the sea because there is no water. And so this was a huge challenge for us in the first survey campaign, because we couldn't really even, enter the site. And so we now had to find a different solution, because this enclosed system where the mesocolumns need the depth and of at least about 1 meter. And we don't have this kind of depth at the moment in the lake at a distance from the center that is reachable by boat, so that we can have citizens visit the floating wetland, take samples and work with us at the site. So it has to be reachable. And so we don't have this system. So and also another really big obstacle for us was the obtainment of permits, which is very bureaucratic. And we even need to obtain permits to survey the mussels, to just enter the lake and actually collect a few samples to analyze it. So we had to find a solution, and the solution is to actually not work at the lake, but work in a tributary river, which is called Volga River. And this also is experiencing pollution, because it is a significant source of nutrients coming from the nearby agricultural fields, so agricultural runoff, but also because the result of the wastewater treatment plants is just thrown into or flows into the river. So the system we are now planning to install there. So you see in this picture, this is where the Volga River is, and this is the site where we're going to install our tanks. So we're going to have about eight IBC tanks where we install the floating wetland and the mussels, and we're going to pump the water from the Volga River into this pump and treat system, and then we're going to pump it out, so the outflow goes back into the Volga River. And this was the best solution we could find. Due to the obstacles that we are dealing with. And we discussed this also with our project officer and of course coordinators later is in the panel, that this could be a solution also maybe for other lakes in Europe that are facing this type of environmental challenges. And regarding citizen science, I just wanted to mention it once more because it's a very strong component of this project. This is run by led by Thames 21, which is a partner in this project. And the citizens will be involved for example, at this site as well. And we will involve them not just for monitoring and sampling, but also for deciding on the plant of this floating wetland, but as well as for empowerment, activate and educate citizens about bioremediation technologies and what is possible to do in this kind of sites, for example, that are facing a lot of environmental challenges. And thank you for your attention.
Thank you very much. And last but not least, we will talk about the phytoremediation, which will be presented by Professor Blanca Velasco Arroyo from University of Berks. So please, Blanca, thank you to find time and to change your schedule to be with us. So please, the floor is yours.
Thank you very much, Aniska. Thank you for the invitation. I'm going to share my presentation. Let's see. Can you see my screen?
Yes, we can.
Okay. Okay, so hello everybody. Thank you very much for this opportunity to share some work that we developed here in the University of Burgos in Spain in the framework of two European projects, Greener and Biosismo. One of them, Greener, is about to finish in 1 month, and Biosismo recently started around 1 year ago. The project coordinator is going to talk later about it. So we are going to talk in a general way about the phytoremediation, and then I will explain what we exactly develop here in the University of Burgos. So these pictures correspond to the river of our beautiful city, Burgos, here in Spain. And this river is very clear, is very transparent, and definitively is very clean. And we have to ask ourselves why it's so clean, because it's a urban river. So the answer to this question of why it's so clean can be in part in the plants that are surrounding the riverside. These plants are mainly microphytes, that are plants that grow in the water, submerged, emerged, or even floating, and they have the capacity to clean these rivers. In this case, the rivers, but we can also talk about different water bodies that we will talk later. So I'm going to talk about phytoremediation in general. Phytoremediation, I'm imagining that most of you are familiar with this concept, is the capacity, the inherent capacity that some plants, even trees, have themselves to take up substances from the water or the soil matrices, and they have the capacity to either sequestrate, stabilize, or even transform them into less harmful and toxic substances through different methods: phytostabilization, phytovolatilization, degradation, rhizofiltration through the roots, etc. So there are different methodologies inside this phytoremediation. So this problem has been raised already for the different experts that have talked today. The problem is very evident in not only in Europe but worldwide. But focusing in Europe, Europe's water bodies suffer from very problematic agricultural pollution. Sometimes insufficiently treated wastewater from cities, and a lot of pollution from industry and space dwellings, according to data from the European Environment Agency. As you can see in this map, in red, we can observe that a lot of places in Europe are extremely contaminated, talking about groundwater or freshwater places. So we need a solution. Especially important is the contamination due to metals and metalloids, and especially in the groundwater bodies in Europe. I raise here an example of one of the metals, that is the cadmium, that this European Environmental Agency has raised concern because it's a contaminant that can be bioaccumulated also in the food chain, and there are some evidences that can cause not only environmental problems but also health problems in the population. So not to be alarmistic, but we have to be aware of this important problem, not only with this metal, but with a lot of metals that can be found due to this contamination. So we have the problem that we are aware of it, and the solution. Okay, we have the conventional remediation solutions that in some cases they work, but are time-consuming, are expensive, and sometimes not ecologically friendly. And currently, we know that we need to focus and to look for environmental. We need to look for sustainable solutions. So we need to stand on biotechnology, nature-based solutions, and embrace the circular economy. And in this term, phytoremediation is a solution. Here in the University of Burgos, we are trying to, in the framework of these two projects that I commented at the beginning, Greener and Biosismo, as part of the work that we develop in bioremediation, we focus on phytoremediation to fight against metalloids contamination. But apart from the plants that are the key players, as I was mentioning, we don't have to forget that plants don't work alone. Some experts already provide very interesting talks on this. And we have to say that normally this phytoremediation is supported by the hidden allies that are the bacteria, the fungi, and the microorganisms that are in the rhizosphere or inside the plants. So there is a collaboration between the good, the ugly, and the bad in terms of these microorganisms, and they play together to decontaminate these water or soil bodies, in this case the water bodies, to transform very toxic substances into less harmful derivatives. Sometimes, in terms of phytoremediation, it's very interesting to look for a group of microorganisms, either bacteria or fungi, that are called plant growth promoting rhizobacteria. They have very interesting functions for the plant, since they biostimulate, providing different substances to allow the growth of the plants. They biofertilize, they bioprotect even against diseases and pests. And in turn, the plants provide these PGPRs the place where they can survive. So this is a very interesting, we don't have to forget about this. They play together, plants and microorganisms. So just to finish, and very briefly, what is done here at the University of Burgos in relation to water contaminated with metals and this phytoremediation in the context of these two projects? Well, we are working with a real groundwater body, a sample that is coming from Belgium, from an ancient smelter industry, and this water sample is contaminated with all these metals and metalloids that you can observe here, that they exceed in an important way the threshold limits permitted by the law. So the objective is to reduce this contamination. We have been studying surrounding macrophytes that grow all around us in the rivers. When you have a walk in your city, you can find easily these species among these 10 species. So we have been testing different assays with these 10 macrophytes. And what we have been doing, we have been doing assays at a microcosmos scale, just testing how they grow in this contaminated water, either with microorganisms inoculated or without the microorganisms, to compare the yields. And we have performed a very big battery of biochemical and also microbial assays, of course, that I'm not going to detail now, but we have observed very important differences that permitted us to select 2 of the species, specifically Pragmites australis and Cirpus orlochoenus, to continue with this. Okay, this is the aspect of one of them that was the candidate and another one that was not performing well. So this allows us, as I was commenting, to select 2 species to perform assays in a higher scale, at a mesocosmos scale. And currently we are with pilot studies, also in combination with other partners of the projects, who are experts on bioelectrochemistry. This has been commented already before. And it's very interesting the performance that can be obtained with the combination of this phytoremediation and bioelectrochemistry. And just to finish, because, okay, the plants can stabilize the metals, even sometimes they can transport the metals to the leaves, to the aerial parts. So what can we do with these plants that are contaminated and are full of metals in some cases? What is the post-harvest strategy? Okay, several issues can be addressed at this regard. We can prepare compost from this, but we have to take into account the law as well. Even another solution is to get some aromatic substances when we have this kind of plants, and even we can precipitate or extract the metals that have been accumulated in these plants. But there are This is a very hot topic, what to do with these harvested contaminated plants that can be treated in another talk. And this is the last slide, just to mention that phytoremediation, apart from this environmental contribution to decontaminate different parts in the soil or in the water, they address different strategies that are in the politics of the European Union strategy, and they contribute also to, for instance, the EU pollinators strategy, and they contribute to increase the biodiversity of the places where they can be grown. So this is all. Thank you very much.
Thank you very much, Blanca. And now we smoothly will go to panel discussion. But beforehand, I would like to ask you the last question. If you know already these all techniques, how much trust do you have in the safety and effectiveness of bioremediation methods? So if you can look as display the last question. And also I would, after this talk, we will send you a little bit longer questionnaire, which will take you, I don't know, at least 5 minutes to fill in. We've kindly asked, to provide us an answer, because for us, conducting such projects are very important as stakeholder engagement, and also your opinion about this solution. So also would like to thank you, Patrick behind the scene, Patrick de Bover, who support us behind the scene with this questionnaire from Perseus, and will prepare the opinion poll. So after this talk, we will send you the link. We've kindly asked to support us and fill in the answer. So now, let's go to the panel discussion, because we have a few questions which we want to discuss. And I would like to introduce and warmly welcome Dr. Thomas Reichenauer from Austrian Institute of Technology. Thomas is a coordinator of BIREM project, also funded by Horizon Europe. I would like to welcome and introduce Dr. Leire Ruiz Rubio, professor from the Universitat del País Vasco, and Leire is coordinator of Symbiorem project. I would like to welcome Lila Otero, team manager at IDENER, and Lila is coordinator of Biosysmo project. And we have with us Giulio Zanaroli, which you already know from the first part of our circular talk. And Giulio representing Universitat de Bologna, he's also the coordinator of NYMPHE project. So as you see, already there is like a few research are going on and conducted all over Europe with many partners from different European countries. So maybe firstly, I will give a floor to Thomas, because not much was said yet about the BIREM project. So if you can now introduce a little bit to your project, what is special about it, and what research you are doing within the project.
Okay, thank you, Agnieszka. It's my pleasure to be here today. Yeah, let me shortly introduce the MIBIREM project. So in MIBIREM, we are focusing on microbial degradation of 3 kinds of contaminants. These are petroleum hydrocarbons, cyanides, and HCH. This is hexachlorocyclohexane. You might know the former pesticide lindane, which is banned now, but there are still many sites in Europe where we can find this as a pollution. And our aim in MIBIREM is to isolate whole microbiomes. We have heard about microbiomes already, how important they are, this network of different bacteria, but not only to analyze them with metagenomics, but also to store them as a whole by freezing them at -80 degrees and providing them for later research and also for remediation. So in MIBIREM, we also are starting with sampling from contaminated sites, and then we end up with pilots. So the aim is to test these microbiomes at contaminated sites to see if we are successful with application of not only individual strains, but also whole microbiomes. So this was in very short words, the description of MIBIREM.
Thank you very much. So maybe I ask you the question which we just asked the audience. Are the bioremediation techniques safe for the environment? Because when you see the answer, 30% that they have complete trust in this, but 50% have some trust with reservation, and 10% said I'm uncertain. So could you convince us that it's safe?
For me, it's absolutely safe because we use nature, and we just try to engineer the nature, bacteria, plants, whatsoever, in a way that we remove and degrade contaminants. And even if we put bacteria at a contaminated site, we have to consider that this is a very special environment, even if there are bacteria that can do certain things like degrading certain contaminants. As soon as these contaminants are degraded, these bacteria will disappear from the site because they lose their food. So if we, as long as we don't use genetically modified organisms, which is of course another topic which can be discussed, I see this as an absolute safe method. Of course, we have degradation products that we have to consider and have to monitor, but this is not specific for bioremediation. This is a problem with any kind of remediation we have, but per se, for me, bioremediation is very safe.
So what the rest of you? I don't know, Leila, Lila, Giulio, who would like to add something to that?
So just, yeah, I completely agree with Thomas. With bioremediation, you usually don't need to add any hazardous chemical. Bacteria may need additional food apart from the pollutants, but usually this additional food is in the form of nutrients that are safe chemicals. And of course, in any intervention that you have to do in an open environment, you also do risk assessment to analyze in detail the different pros and cons of the technology, and usually bioremediation plays results good in those risk assessments.
Leila, I think you are mute. You have to unmute yourself.
Now? Yes.
Yes,
okay,
we can.
Thank you. For example, in our case, we were concerned about these issues. That is why we have had not only a risk assessment, as Lila comments, we also have one manager for ethical issues that she is in charge of checking all of the steps that could be concern us about ethics. As, for example, Tiziana shows, we are going to use mussels, so we have an evaluation of ethics about using these mussels, and so also for this kind of genetic modification of the biological the microbiota that we are going to test in a lab. We are concerned about that, so we have our internal ethical committee evaluation previously of going further in the investigation. So we try to go step by step and try to realize if should be some ethics problem or some risk on this kind of new bioremediation technologies.
Thank you. Giulio, would you like comment on that?
Yes, I mean, I can only say that I agree on the opinions of the other coordinators that bioremediation can be considered a safe approach, a safe solution, of course, after, let's say, all the assessments anyway have been carried out in a very rigorous way. And here, of course, we should consider mainly the use of non-genetically modified organisms, which are the, let's say, players that we use now, and maybe in the future also others. And of course, the safety must be related. I mean, in bioremediation, we always take care of the products of these degradation processes that may or should be complete, let's say, and fully degrade chemicals. They can form intermediates, of course, or byproducts that typically are assessed in terms of chemical and toxical features to make sure that, let's say, the safety is, I mean, the process is safe.
Thank you very much. There is one question about the scaling up. It also was my question to you, how we can scale up this solution, because so far I think they use or more or less on some limited areas. But can we commercialize? There is, I think, some examples that it goes to the full commercialization, yes, some of these techniques. So how you can refer on that? Can we scale it up? What is the barriers? And, you know, how do you see this problem or topic issue? Thomas, you want to start?
Yes. Yeah. Of course, scaling up needs always pilots. I think, and this was what is implemented in all of our projects. So it's always the aim to test the different technologies in a pilot. I think the difficulties are that we would need not only one pilot, but maybe several pilots, because there are each contaminated site is different and needs special planning, special treatment. So if a pilot is not so successful, then it should be not a KO criteria, but there should be the possibility to do repeated on other sites and other pilots. And generally, I think the difficulty is the interaction between different stakeholders and different knowledge groups, let's say. And this makes it also, or let's say this is a challenge in scaling up these technologies, because they are very know-how intensive and they need know-how from many different experts. So this makes it quite complex to scale up.
What is the other's opinion on that topic?
For example, in our case, we are going to try to go at the end of the project to a couple of full-scale approach. Of course, first we have in the middle of the project this pilot scale, but I believe that sometimes perhaps, and that is why in our project we have a strong belief in citizen science. In order to go to this commercialization, we need people believe in us. I mean, people believing in that bioremediation could be a key technology for recovering soil and water. So this is our main goal, and I think that this kind of initiative in which all the projects we meet together and we speak together are great to change this thinking of the people and start growing this believing in the importance of bioremediation. So our key things to do is to try to do our best and to share all the results that we have, because there is not, let's say, a recipe to remediate all the soil in Europe, as Thomas says. It's quite challenging, and we have to speak a lot together in order to go fast.
Giulio, Lila, would like to add something to that? Giulio, you want?
I can say yes, I agree that probably the major problem is, let's say, the replicability under on different sites, because bioremediation is not only, let's say, the technology that you apply, but also the way you apply to a site which has its specific features in terms of not only pollutants, but also hydrogeology, etc. So it requires, let's say, the integration of different competences, not only the ones related, let's say, to biotechnology, but really the integration of different competences to make the process be effective in the site-specific conditions. So this is probably, let's say, an important aspect that has to be considered in order to, let's say, promote then efficient implementation.
Lila, would you like to add something?
Yes, just to add to that. I think that is also a part of the replicability. I think it's also important to know under what conditions bioremediation is feasible and under what conditions is not, because there are conditions where it's not, of course. But in those cases, we can always think about combinations of different technologies, which is also important. And also I think that for the perception of the not only citizens, but also for the administrations or companies that have to apply these technologies, we have to be clear that sometimes bioremediation is feasible, but it takes more time than other technologies. But it works. So it also depends on the needs of the specific case study.
And from the policy and funding perspective, do you think that it's enough already and enough funding for such project, or maybe you think that there will be more support and it should become to the more mainstream research bioremediation? How do you find it? What do you need to really kind of speed up your research from this policy or funding perspective?
So should I start again?
If you want, please.
Yeah, okay. Yeah, of course. I mean, there never can be enough money because there are enough questions that we should answer. So in principle, I would say the more support, the better. I mean, I think the bioremediation approach is quite comparably new. I can talk for Austria. I mean, in the past, remediation was dominated by construction companies and by geologists, and biologists are a new group in this area. So there is no, I would say, not such a lobby as the older groups have. So I think it's in many cases more difficult for to convince politicians or decision makers in political positions that this is a good approach and this should be followed, and there should also be money for that. So I think there is still some potential to improve the support from the governmental side. But I guess this is very different from country to country. So this is very country specific, let's say.
What is the other panelist thought on that?
I agree, and also I think that specifically because of the difficulty of replicating the technology and really needing to study specific cases, and also going back to what Leire mentioned, I think that it's also needed some coordination from above to make sure that results of different projects are shared between us, and that we can really make we have the resources to make these results to matter and to use data from that is available and that can improve the replicability in different sites.
Leire, Giulio, you want?
I mean, this is easy. It's like telling to my daughter that she has enough chocolate. No, never. So a researcher never. We don't have enough money never. But I really believe that this new initiative in this new Horizon Europe goes in the right directions. But my concern is that if they are going to still in the time making this effort, because now I believe this mission, soil mission, water mission, this CL6 are very nice, very interesting, and give us quite good support. But if they suddenly cut this off, we are going to lose a lot of time, a lot of resources, and a lot of interesting things that we are doing right now.
Giulio, you want to comment?
Yes, I mean, I agree with this last point. There is need to keep, let's say, investing on this because otherwise there is the risk that, yes, we lose a lot of, let's say, work and resources before we can make the transition, let's say, to these technologies completed.
Thank you. And maybe the last question, because we are run of time. There is like some question from the public about Asia and Africa, because mostly the project which was presented are going on in Europe. So do you know about some project which is also on outside of Europe? Yes, I know that Electra, I think, was cooperated with in China. Yes, so I don't know what is going on in this topic on the other continents in other part of the world. Are you familiar with that?
So from my side, I've unfortunately no cooperations currently with other continents in that respect. But I know that China is doing a lot, and also I've also seen papers from South America and also from Africa. In my opinion, or in my vision, it seems that I think that that bioremediation is a very good technologies for countries, also for developing countries in other parts of the world, because it's a technology that can be easily applied. What you need to know is you need to know or have a lot of know-how how you do it, and this but it does not need much technology if you know how to do it. So I think these are in principle good technologies, and as far as I know, there is a lot of things going on also in other parts of the world. But as I said, I have currently no cooperations with countries in other parts of the world. But maybe other colleagues can say more about that.
Anyone? Julia? Please, Julia.
I had some experience of collaboration with Chinese researchers in the LETRA project, and yes, the feeling is that they are pushing not only as researchers, of course, but also, let's say, in terms of funding on this on bioremediation, and also, let's say, on the scale up and pilot testing of these technologies.
So yes, please, Leila.
No, I was for example, our international board. There we have some experts of other areas: Chinese, Indian, Australian, and they are also in similar move than us. Luckily, also we have some partners involved in or had been involved in previous projects in collaboration with Africa. But I think that we still have not so many programs to exchange our knowledge to less developed countries. Perhaps this also should be a point.
So thank you very much. As I see, it's 12 o'clock, so we should finish now, unfortunately. But there is, I think, much more to say, and it's still on the beginning of the development roads, I think, the bioremediation processes. So please follow our websites. Of course, I'm talking for NIMFE project as I'm responsible for communication, but also there is Agile Horizon project, which I also recommend you to see how we proceed with our research. So once again, thank you for being with us. The webinar is recorded, so it will be available later on the European Circular Economy Stakeholder Platform webpage. Thank you and have a nice the rest of your day. Bye.